Hello and welcome to Learning Science Daily. In this video, I will be telling you about how to perform the analysis after DPPH assay, and I will also be discussing about how to calculate the percentage DPPH radical scavenging activity. So this is the data that I have got after performing the experiment. So I did my experiment in a 96 well plate, and you can see the highlighted wells are the one where I did my experiment. So in these three wells contains only 200 microliters of methanol, and the second, if you see in the second column, these three wells contained 100 microliter of DPPH added with 100 microliter of methanol. And from this D to H, I have added 100 microliter of DPPH with 100 microliter of my compound and standard. So I have written here in which wells I have added the compounds and in which wells I have added the standard molecule. I have taken ascorbic acid as standard in this experiment, and if you see, these are the concentrations. These are the 5 different concentrations that I took for the compound as well as for the standards. These are in micromolar concentration. So now we have to perform the analysis. So you just do, the first thing you have to do is to copy this whole data into a new sheet. So as you can see, I have added the data in sheet 2, and you can also rename this as analysis. Okay, and we can rename sheet 1 as raw data. Okay, so in the analysis sheet, the first thing you have to do is to normalize the data. The normalization is done by subtracting the absorbance value of your compound or your standard or your control with the methanol. So here I am performing the normalization for this blank. This is our blank. Yeah. So you have to use a formula. This reading subtracted by this methanol reading. Yeah, so here you will get the normalization. So I will just drag it down, and yeah. And similarly, you have to perform the normalization for all of the different concentrations. So you just have to put the same formula by subtracting the reading and then drag it down. Okay, so the normalization is completed now. Now we have to calculate the percentage DPPH radical scavenging activity. So you know the formula for, so I am telling you the formula for calculating the percentage RSA. So for calculating the percentage RSA, you have to subtract first the value of your control, this thing, with your sample divided by control into 100. So here we can calculate the percentage RSA. So this will be equals to, you have to put a bracket and then you have to subtract this value divided by your control into 1000. Yes, so here you get the percentage RSA. This is for 100 micromolar concentration, and similarly you have to calculate the percentage RSA for all the concentrations. And here I am again using the same formula for 50 micromolar concentration. Yeah, so it's done. I have calculated the percentage RSA for all 5 concentration, and remember I have done this thing for this compound. This is not of the standard. This is the compound percentage RSA at different concentrations. Now we will calculate the average. So we will just put the formula here. Okay. And we will just drag this. Okay. And we will also calculate the standard deviation, because if you want to plot a graph, you need these values. Okay, so now we can add a graph here. So you just have to write these values, paste values, transpose. Okay, so this is the concentration, and here we will write the percentage radical scavenging activity. So we will just copy the average value and paste the values here. Okay, And now we can draw a graph. So you just have to select these values, insert, then you have to, like this, you have to insert the graph like this. Yeah. So I'm not formatting the graph here, but if you want to incorporate the data in your paper, so you have to prepare the graph accordingly, but this is how it's done. So I am not going to show you how to calculate the values for standard, but yeah, for any compound or whatever compound you are testing, the calculation will be done similarly for all of them. So I think the analysis is clear to you now. Thanks, and please subscribe my channel, and if you have any doubt, comment in the section below. I will be really happy to help. Okay, then see you in the next video.